we talked about this last year. We talked about SC3, these new ingredients. So let's talk about this new ingredient. Now, to understand the magnitude, I think, of what we have discovered and what we have right now, let's just put that into the global context of how we developed all of this. As you remember, it started with, and here I just want to put this again in the global context. When we discovered, it's not like we brought a new or the novel best antioxidant or the novel best anti-inflammatory compound or plant. What we did is that we developed an entirely new concept in health and wellness, and then a product that really tapped into this entire new concept. So it's really quite something that was brought to the marketplace. And it was done with the discovery of Stem-Enhance, AFA, and then its extract or concentrate Stem-Enhance. Then we started to study different plants that led us to the discovery of fucoidan, for example, goji berry, medicinal mushroom, different compounds having different kinds of effect on stem cells, and it led to the development of SC2 and MigraStem. Now, we continue to do that work, and we search with plants in other remote areas of the world. So I was in contact with a team. It was a biochemist and a pharmacist, and they went to Bolivia, they went to Papua New Guinea, they went to Madagascar. They collected many, many plants, documented many of these plants with the indigenous local use of these plants. So when they were talking about all their adventure, I asked them what was for them a very, I would say, silly question. I asked them, is there a plant that when you ask these local healers, is there something that is good for everything or for many different things? And they kind of laugh, well, not laugh, but they look, they smile, and obviously such a thing does not exist. You don't have something that is good for everything. It doesn't fit the traditional scientific model or medical model. And then I went on to share with them a little bit more what we had as a concept, the role of stem cells in the body. I gave them a copy of my book, Cracking the Stem Cell Code. And when they started to sink their mind into all of this, they came back and they realized that the question was not futile. If you have something that has an effect on stem cells, it would lead to a broad variety of benefits. So they went back into their plants, and a few weeks later, they gave me a call and they said, I think we have something from Madagascar. And the plant is a subspecies of aloe called Aloe macroclada. So there are many, many species of aloe, as you know. There are about 65 species that are endemic to Madagascar. So there are 3 families in Madagascar from generations to generations that have made this product locally. It's a product that you can buy. This is what it looks like. They take the gel, they char, they burn some parts of the plant, they use the ash, they roll that with the gel, they make these little beads, they dry them under the sun, and when the way that they talk about these little pellets, these little beads, these little pills in Madagascar is that if you have any kind of health problems, just take these pills and you will do much better. And if you keep taking them, you'll never be sick again. So that's, of course, it's kind of a fruitcake kind of statement, but that's what we're after. We're not after something that helps everything. We're after something that has one effect, to support the natural release of stem cells, because we know what this impact will be in the body. So I'll come back to the data that we've had on these plants. We tested them, and we got something that was actually better than anything else we had seen before. So when we got that kind of data, then we started to dig a little bit further as to what do we have to do to really develop this product and bring it to the marketplace. And that's when really this whole story became what, to me right now, is probably the greatest adventure that I've been associated with. It is a plant that grows in this kind of environment. So this is about within 2-hour driving radius outside of Antananarivo, the capital of Madagascar. So we drove to go to this place 2 hours. We parked the car along the road. Then we took a small trail and we walked for about 1.5 hours. And then we start to see houses like those that you see, and they're everywhere. Now, there's no roads, so where are they coming from? And they essentially dig the ground, they make clay bricks that they bake, and then they build their houses with these clay bricks. And I can tell you, they're extremely clean. I mean, it's quite, I don't know what's the right word. I was impressed. I'd never seen something like this. So extremely well organized. All the fields that you see everywhere around, it's rice fields, beans, peas, flour, corn, wheat, all kinds of things. They grow everything that they eat in these fields. And this is, again, hours away of walk, in the mountains. That's where aloe macroclada is growing. That's a little bit what the houses look like if you go closer to some of those. So when you think about going that far away in the mountain to bring back these plants back to the road, that means that they harvest the leaves, they put that in a bag, they put the bag on their head, and they walk for a few kilometers back to the pickup truck, and they carry like this at this point today to prepare for the launch of SE3 and then the sustainability of the whole project. They carry 2 to 5 tons of these leaf every month back to the road. So there's no road, so it cannot be done even on a horse. So we are going to develop, and I'll come back to that in a few moments, but we're going to develop roads and ways to improve the whole supply chain. Here's a picture of John, the fellow biochemist, who, after I shared the data with him, quit his job and decided that this was also for him a great adventure. So now he is in Madagascar developing the whole infrastructure to make this possible. At the other end, we have Fabrice, who are our contact in Madagascar. He's Malagasy, so he is our local support and doing really a fantastic job to the two of them to develop this whole project. Now, one of the most delicate, or I should say, daunting, one of the most difficult part of the whole project, and you need to visit Madagascar to realize how complex that is. And it was to connect with all the landowners, all the people who own lands on which this plant, Aloe macroclada, was growing in the wild, because anybody can go, in theory, and cut this aloe, harvest this aloe. Now, it's not true, because there are regulations in Madagascar. It is a very unique biodiversity. So you cannot just get anything from the ground and use it. You need license to harvest, you need license to carry, you need license to export. But in Madagascar, like many places in Africa, most people will do it without getting the proper license, and everything is done sort of by paying under the table. So when we arrived there, we basically decided that we were going to do the things right. So we went to see the government, and we started to apply for all the licenses. And at first they were surprised because nobody had done that before. So we showed them how to fill these forms, and they learned the process. So right now we are in a place where we have all the licenses, everything has been done to really do everything right. And in that way, we connected with all these landowners, and we developed an association, knowing very well that the day that we launch this product, other people may want to go to Madagascar and try to harvest these plants as well. So we've developed a really, really tight networks of all these landowners, telling them what they were associated with in terms of the project leading to the launch of this ingredient worldwide. They're understanding what they're coming part of, and they're very excited to be part of this adventure. So, but whenever we want to tell them, okay, this week we are harvesting, there's no cell phone down there. Or if they have a cell phone, there's no dot, there's no bars. So it doesn't matter. So when it's time to harvest, they need to jump in their pickup truck, drive 2 hours, walk an hour and a half, and tell them, we're harvesting next week. They need to walk back, jump in their pickup, going to the next place. So it'd take about 2 to 3, 4 days of driving to tell them we'll harvest next week. And now if for whatever reason the truck has a flat tire or something happens that they cannot harvest, they need to go back for 3 days on the road to tell them, no, it's 1 week delay. You get the gist here of the kind of logistics that needs to be put forward, and what needs to be done to keep this association extremely tight. It is really something that I had never experienced before. It's kind of a different world in order to do business, but at the same time, it's so exciting because it's all done by trust and relationship. This entire association is strictly held by eye-to-eye contact, handshake, and the trust that we have developed over the past 2 years with these people. It's the first time I'm involved in something like this. It's really beautiful. So here's a picture of the association. This is a picture of Aloe macroclada as it looks in the wild. Now, one thing that we had to do before starting this whole project was to evaluate how much we have of this plant naturally available. Because if we go in the wild and we start to harvest this plant, we could have an impact, a biological or ecological impact. So we hired a local university to go and do a survey of all this region around Antananarivo. And it was, the survey was done and established how many of these plants were already available. And there's enough, but there's not enough to be able to make this harvesting sustainable without replenishing what is being taken. So we started a project where we collect seeds and then we plant. We have a nursery in various regions where we grow small plants, and after that we put them on our heads and we walk back in the mountains and we replant them in the ground. So we have so far in the past 6 months, roughly 6 months, we've planted 12 to 15,000 plants, and the plan here is to go to about 100,000 plants that will be planted all around. It will take about 3 years before these new plants are harvestable, but nonetheless, it gives an idea here of the overall commitment and the overall project. So it took 1 more year compared to when we wanted to first launch the product last year, but it gives you an idea of why it took a little bit more time when we realized that just to connect with the people to do the harvest, the cell phone doesn't work, and then a few other things, we just realized, wait a minute here, we just need to really make sure that when we start, that processing chain, that supply chain really works very well. So here's a little bit what the data is looking like. This is what we got from these pellets. So 1 dose is normally 3 of these pellets. So we went back in the lab, gave 3 pellets to people, and we counted the number of stem cells. What you see down there is the curve that we used to have with AFA. Now, a good way of having an idea of how many stem cells are released in the bloodstream after the consumption of something, you look at the curve, on the area under the curve. So see the curve here and look at what is under the curve, and look at the dark line with the pellets and see what you have under the curve. And keep in mind at 3 hours, the number of stem cells are still increasing. So all we can say here, we cannot quantify because it keeps going. All we can say is the amount of stem cells released in the bloodstream are far, far in excess from what we had originally with stem NNs. But we also have another thing with the product, is that as we started to look for other types of stem cells, we also found that the allomacroclada also supports the release of what we call endothelial progenitor cells. These are cells, stem cells, that have already started to specialize into one area, and it's to make new blood vessels. And it's something that we all know that we need good blood supply, good blood circulation. We all know this. Nevertheless, it's something that we very rarely talk about. A product like StemFlow is still today a unique product on the marketplace. Proper blood circulation in capillaries is absolutely paramount for your health. If you don't have good capillary circulation in your organs, you cannot nourish all the cells with nutrients, oxygen, vitamins, minerals, amino acids, new cells to repair, immune cells. You need good circulation. That's what these cells are doing. Whenever they circulate in your body, if they find a tissue that is lacking proper oxygen supply, they settle there and they develop new capillaries. So it's very, very important for sustaining, supporting optimal health in the body. Now, I don't have this data to share here right now, but we did way more than just that data. By the way, this data right now has been written for publication, and the paper was accepted yesterday. So this should be published in the scientific literature fairly soon. Now, all this data, the reason why we don't publish all this data, I'll talk a little bit more about this tomorrow, is that we're also lumping into a fairly big research project that is a multi-center project in many countries that is going to test about 600 to 800 people. And with that data, we can file claims in the EU, elsewhere in the world, claims that will be like, this product, SC3, supports glucose metabolism, vascular health, whatever the data that we develop through stem cell release. And if we have this data based on non-published data, non-published information, then we get an exclusive in certain countries on that claim for a number of years, so that we could be, for a number of years, the only product who can have that kind of claim. So we're keeping some of that data unpublished, but we did more than just testing these pellets. Knowing how these pellets, these little pills were made, we essentially derived other extracts of allomacroclada that we tested in the lab. And what we have right now with a dose that is about half of what these 3 pellets were, we have data that brings us with an increase actually close to 80%. But we'll see that later on. Okay, so this is the effect of allomacroclada on stem cells. Now, we added another part to this whole story. If you put back in mind this overall picture, we have mobilization of stem cells, circulation of stem cells, migration of stem cells. They're different. So you can look where we're going, but for now, stay with me. Mobilization, circulation, migration. Thank you. And then cells migrate in the tissue, and then they proliferate. When one stem cell enters a tissue, it will proliferate and become anywhere between 1,000 to, let's say, 5,000 cells. Well, there's a difference between 1,000 and 5,000. If you're good in math, there's a 5 times difference. So if you go from 1,000 to 5,000, there's 5 times more repair power, repair potential. So right now, to support that process of proliferation, we have dermostem in the skin. But you cannot put dermostem on your heart or on your brain. Doesn't work. So what can we do to support this process of proliferation everywhere in the body? That's what we added to this bulk product of stem cell release. We added this other part, which is proliferation. And that taps into the whole science of telomeres. And I won't go into too much. This can be very complex. I will simplify it. So if we all go back into our past class of biology, this is a very simplified diagram of cellular division. You start with one cell, then the DNA duplicates, because if the cell is going to separate, you need to have the same DNA in both cells. So the DNA separates, and then you get the whole process that gives us, at the end, 2 identical daughter cells. The focus here is the second step, or the first step, the second sphere, DNA replication. You need to multiply to make a copy of that DNA. Now, if we look at this through this kind of diagram. So here's a cell that has all these chromosomes, and at the end of these chromosomes, we have segments that are called telomeres. When the time comes to make a copy of that DNA, what you need to do is that you need to unzip the DNA, and the DNA will sort of duplicate itself. So if we look at this with a little bit bigger schematic representation, in order to unzip this DNA, the cell, now, it's not really happening that way, but imagine like little arms in the cell, you know, grabbing the ends of these chromosomes. There's little handles. So the cell takes it, holds the chromosome, unzips the chromosome copies itself, and at the end, the little handles just don't let go. They basically clip the part that they were holding, and then the cell goes and continues its process. Every time that the cell divides in that way, it leads to a clipping of these telomeres. Now, the body, stem cells, have enzymes called telomerase to keep the length of those telomeres. So in your bone marrow, your telomeres do not shorten. They maintain themselves during your entire life. But when the cell leaves the bone marrow, gets into the body, in your tissues, the clocks start ticking. And now those telomeres start to shorten. So the initial length of those telomeres and the ability to maintain them as much as we can is going to determine at the end in your tissue how far they can go into that multiplication. So we go back to what we were saying before. There's a big difference between 2 and 10 in terms of your ability to repair. If one stem cell becomes 2,000 cells or 10,000 cells because it has more ability to multiply, it means a big difference at the end in your overall capacity to repair. So keep that in mind. Here's a little schematic representation. There's a study that was done, and you need to really keep that into its proper context. I'm trying here to really simplify it as much as possible. We need to keep this data into its proper context. This was never done in humans, and you will understand why, because this was done on animals that were genetically modified. So they tweaked the gene that makes the enzymes that keeps those telomeres long. So when they let these animals age, what they see is that the telomere shortens much more rapidly and they age much more rapidly. So, and the signs of aging is lipids under the skin. You know, as we age, the skin becomes much thinner. Humans developed glucose intolerance, insulin resistance, bone density decreases. Many things are associated with the simple process of aging. Now, they developed these models of engineered mice so that they could also reverse this process and reallow the lengthening of the telomeres. And when they did this, they see the reverse. As the telomere lengthens, those signs of aging reverse. And from this, the whole explosion in the media went and saying, this is the fountain of youth. It's true, but we really need here to just stay calm. Just stay on your chair. It's okay. Just take a deep breath. We are not biologically engineered to have no telomere, so there's nothing to switch back on. It's sort of already on. So it just shows the full extent of what happens if we can help telomere maintenance, if you want. That's really all that study is showing for us. When we do this in humans, and why I'm saying all of this is that there is one plant that has been documented with the ability of enhancing the activity of the enzyme that maintain telomeres. The studies are showing that this plant is not going to make your telomeres longer, but it's going to maintain their length. So if you maintain their length, you basically give more longevity to the cells. To an animal, what we see when we give that plant to an animal, all these signs of aging, a decrease in glucose tolerance, increase in insulin resistance, decrease in bone density, decrease in subcutaneous fat, all of it is reversed. By reverse, I don't mean extremely reverse. I mean those signs of aging simply start to get better. So at the end, these animals do not live longer, but within their lifespan, they have a much greater health span, which at the end of the day is really what we're after. So what we have here is that we release stem cells, we support their circulation, we support their migration with migrostem in ST5, we support then their proliferation with astragalus, that is part of SE3. So we support the entire process to give just, again, more power to this support of stem cells in the body. So what we have with SE3 is the 3 compounds that you already know having an effect on stem cell release: Steminence, Allomacroclada, Fucoidan. I will come back tomorrow to talk about the study that is going to document the effect on these. And really, it's a study that I've never seen anything like this done with dietary supplements. And added to this, another component, which is an astragalus extract to maintain telomere length. We have a curcumin, turmeric extract, curcumin, because curcumin has been documented in a number of studies to support the longevity of stem cells once they've been released from the bone marrow, to protect them in various ways. And the literature is not super clear, but there's a lot of papers just basically sharing that or showing that curcumin supports the life of stem cells outside of the bone marrow. So we included in the product, and cordyceps for its effect in terms of the synergy that it has with the other ingredients. So essentially, that's the formula that we have with SE3. That's the formula that Ray mentioned before has been tested by a number of people. It's the 3rd generation of SE3. We got great results all along, but I think that with this latest formula, this formula that we're just talking about right now, the results that have been coming back has been so far, I think, better than anything that I would have expected. And I think that we have some testimonials. Some people are going to share their experience because we've shipped out a number of bottles. People have had a chance to try them. So to tell you a little bit more about this, do I have Dawn? Give hand to Dawn.